today. We're very grateful to the SADS Foundation for hosting the webinar, and enormous thanks also go to Tenaya Therapeutics, who are sponsoring this educational event. If you have any questions, please will you type these into the chat, and they will be addressed after the speakers. The webinar is also being recorded so that you'll be able to obtain a copy afterwards. Erin, please could I have the agenda slide, followed by the bio? Our speakers today are Professor Elijah Behr and Dr. Whit Tingley. Professor Elijah Behr will speak about Genetics 101, ARVC and HCM. Dr. Whit Tingley will provide us an overview of Tenaya's clinical trials for HCM and ARVC. Dr. Tingley joined Tenaya as Chief Medical Officer in December 2018. Previously, he served at Cytokinetics as Vice President of Clinical Development, and Cardiology, and at Genentech as Therapeutic Area Lead for Cardiovascular and Metabolism Research and Early Development. Whit formerly was Adjunct Associate Professor of Medicine in the Cardiology Division at the University of California, San Francisco, and cared for patients as an attending cardiologist in their cardiology faculty practice. Dr. Tingley earned a Bachelor of Arts degree from Brown University and his MD and PhD degrees from the Johns Hopkins University School of Medicine. He completed internship and residency programs at the Johns Hopkins Hospital, a cardiology fellowship at UCSF, and postdoctoral research at the Gladstone Institute. I'm now pleased to let you know some more about Professor Behr. Professor Elijah Behr is an international expert in the field of arrhythmias and is Professor in Cardiovascular Medicine at St George's University Hospital, London, where he directs the Cardiovascular and Genomic Research Institute. He is a consultant at St George's University Hospital and the Mayo Clinic Healthcare in London. He leads nationally and internationally reputed clinical services dedicated to patients with arrhythmias and patients and their families with genetic risks for arrhythmia, cardiac arrest, and sudden death. He has co-written multiple international guideline documents and published over 300 articles in peer-reviewed journals. He is past president of the Association of Inherited Cardiac Conditions and the founder and first chairperson of the European Cardiac Arrhythmia Genetics Group of ARR. He is also first chair of the Predisposition and Screening Community of the Genome England Research Network and advises NHS England clinical reference groups for cardiovascular and genomics services. He leads the NHS England Coronal Sudden Unexpected Death Pilot Programme. Professor Behr also leads a research group with interests that include the genomics and prediction of sudden death risk, drug-induced arrhythmia, sudden arrhythmic death syndrome, SADS, and sudden infant death syndrome, SIDS. Also, channelopathies including long QT and Brugada syndromes and arrhythmogenic cardiomyopathy. And we're honoured that Professor Behr and Dr. Tingley are joining us today. So before we start the webinar, please just be reminded that you can ask questions by typing them into the chat feature, and the webinar is also being recorded. I'm now delighted to pass over to our first speaker, Professor Elijah Behr. Over to you, Elijah.
Thanks, Anne. Really appreciate the introduction, and also appreciate the use of the photo from about 20 years ago. As you can see, I have a bit more grey hair since then. So I'll be giving just a beginner's background overview of genetics and relating that to the two conditions that we're going to talk about today, ARVC and HCM. If we could forward the first slide, please, or the next slide. So in every cell of the body, cells making up the organs and components of the body, most of them will have genetic material inside them. The genetic material programs that cell to be what it is. And it's all held inside the nucleus inside the cell. as well as in some other locations. But most of what programs us and that we pass on to our children, what we receive from our parents, is held in the chromosomes that are held in the nucleus. These are made up of DNA, the material that makes up our genetic program. The DNA is broken up into a number of different segments. Particularly important to us today are the gene segments, the segments that that are responsible for producing the building blocks of every cell of the body, the proteins, and are also the subject of potentially disease-creating processes that we're going to talk about today. So next slide, please. If we have changes in that programming code that is responsible for the gene, then we could potentially cause changes in the product. Now, not all variation in the genetic code that we find in humans and in people who have diseases like cardiomyopathy are responsible for their condition, but we have established that some are really important. And what is really important is when that change in the gene results in no protein being produced or not enough being produced, or the protein doesn't function normally the way it should do, and therefore it doesn't lead to a normally functioning heart. And that can therefore result in heart conditions. These are called genetic variants and have been called mutations in the past, so we'll use the term mutation now. Now, we can receive those from our parents, as I've mentioned, and pass them on to our children, but in a small number of cases, they'll be also caused by exposure to things like radiation that's in the environment. And that can cause something called sporadic or de novo mutation in an individual. Next slide. Arrhythmogenic right ventricular cardiomyopathy, ARVC for short, is one of the important conditions we're considering today, and mutations in the PKP2 gene result in lower levels of the protein plakophilin, PKP2, as coded for by the gene. This is a really important protein involved in the desmosome. Now, the desmosome is a component, as you can see from those diagrams at the bottom, that links together cells that lie next to each other. So these are cells that are in the heart called myocytes. When myocytes pump, they contract, much like your muscle in your arm will contract in response to you flexing your biceps. The heart will do the same in response to electrical impulse to pump out to the body. When those cells pump, they create force, and they need to make that force work with other cells. The cells are physically joined together. What happens when PKP2 is missing is the desmosome is faulty, and this can cause breakup of the connections between the cells. The desmosomes become damaged, and this in turn can lead to replacement of heart cells with tissue that is abnormal. That can be scar tissue and even fatty tissue. We call this fibrofatty infiltration, and it's one of the important signs of ARVC. Next slide, please. And why is this important? Well, because this can then lead to electrical short circuits in the heart that can cause arrhythmias, rhythm disturbances. And these rhythm disturbances can cause potentially sudden death. And that's an important potential risk for ARVC that we endeavor to treat patients for. There's also the potential for that scar tissue to damage the heart in the longer term, to cause heart failure, meaning a weakening of the heart muscle in being able to pump efficiently for the requirements of the body. And that can cause symptoms such as breathlessness, swelling of the abdomen, and swelling in the lower limbs because of accumulation of fluid. And we really aim to try and prevent these conditions from occurring due to PKP2 disease. And as you can see from the title, there are not a small number of people around the country affected by PKP2, there may be over 13,000 people affected by it. Next slide, please. In terms of what we do currently to try and avert those potential side effects of the progression of ARVC, there are medicines, for example. These can help reduce the risk of arrhythmias, meaning heart rhythm disturbances occurring. These can also be used to try and reduce the progression to heart failure in the longer term. But these are not necessarily fully effective, and there can be side effects from medications. There are also device treatments, such as implantable cardiovascular defibrillators, and these are very useful for preventing people who are at high risk for arrhythmias and sudden death from having life-threatening events. But they are subject to risks, including complications, inappropriate shocks, and some of the anxiety and impacts on quality of life that can arise from that. There is also ablation therapy that can be used to treat the electrical short circuits in the heart directly, but these are not always successful, and there's often long-term recurrence. But there's also a need to restrict exercise, because we know that exercise, particularly endurance, high-intensity exercise, can not only precipitate liver problems, but cause the fatty infiltration to occur over time, and that in turn has a negative impact on quality of life in general, particularly for our younger patients. And so we really need that option to prevent the onset of disease, to prevent disease progression, and to improve quality of life and the disease state in general, that goes beyond just the standard care we have available at the moment. Next slide. I also want to touch briefly on hypertrophic cardiomyopathy, which is another heart muscle disorder that's important and more frequently seen in than ARVC. As you can see, the numbers affected by this particular subtype of hypertrophic cardiomyopathy due to the gene MYBPC3 having mutations in it, that's estimated around 27,000 people in the UK alone. And that's responsible for around half of hypertrophic cardiomyopathy that's inherited in families, and can be very important in childhood onset hypertrophic cardiomyopathy as well. Now, rather than the heart muscle being replaced here by fatty infiltration, the heart muscle becomes thickened and enlarged, and is impaired in its function. And that can lead to symptoms such as breathlessness or chest pain. It can also lead to arrhythmias and the risk of sudden death as well. And so it has very similar social and psychological impacts to ARVC. Thank you. Next slide, please. So in terms of potential clinical trials that can address the additional needs that we have for treating patients with ARVC and HCM, they are important, but they do have particular demands that they're going to place upon patients who are interested in taking part in. And today we really focus on the ARVC trial, because that is just launching in the UK currently. The questions you'll need to consider are, will the study actually fit into your life? Can you manage it in your lifestyle as a working person, for example? And what expectations do we have about the outcome of the trial? The initial trials are going to be focused on safety and effectiveness of the treatment, and may not necessarily result in any evidence of a benefit for individuals who participate. So there will be more of an altruistic requirement to take part in the trial, because there will not only be some challenges to your lifestyle in terms of participating in this trial, but there will also be potential risks as well. I'll hear a bit more about that from Whit. There are also the benefits that may arise from that, and I said that there are the benefits that may be more altruistic than personal in this circumstance. And you may also want to have some degree of trust in the clinical team looking after you to begin with. And therefore, if you're coming to a trial from outside of your normal clinical team, that may be a challenge for you. You may also need a lot of information, and you'll get that from the people conducting the trial, like myself. But you'll also need to have others giving you support information. There is that additional information out there, and organizations such as SADS UK will also be there to provide some support as well. Next slide, please. So patients have a really important role to play in drug development, not only to participate in the trials themselves, but also to inspire the trials, to help us decide how best to develop trials and what we should be focusing on. So even before trials begin, there are the preclinical phases where we need to know, well, what is important to the patient and to family members, where should we be focusing our trial development. That is not only around advisory groups, but also about patients being willing to join registries that we call natural history studies that tell us how the disease progresses and therefore where we should be intervening with clinical trials. And then we have trials phases 1 to 3 that are all about initially showing that the treatment works in terms of affecting the heart, that it's safe to do so, safe within reason, because there will always be some risk for any new treatments or for any treatment. But then our best to demonstrate that it's going to be effective at preventing disease and disease development in individuals. And so not only is there the interest of participating, as I mentioned, but also giving us your feedback on clinical study design and to help educate others, not only other patients, but regulators, about how therapy, so this is the FDA or the MHRA, the regulatory bodies that are important to deciding whether you or your family will get gene therapy in the future. And then finally, in phase 4, there are the longer term follow-up studies to really demonstrate the long-term success of these potential new treatments. And patient participation is clearly vital to ensure that that happens, and to understand the barriers to uptake of gene therapy trials and of gene therapy itself beyond the trial in NHS and in other health services around the world. Next slide, please. So now I'm going to hand over to Whit, who will tell you about gene therapy.
Yeah, thank you so much, Dr. Behr, for joining us today and sharing your expertise on genetics and heart disease, and sharing how people affected by these diseases and their families can participate and help us all create new, better options for the future. I know at Tenaya Therapeutics, we very much rely on patient participation. Clearly in the clinical trial format, both in trials of drugs and in natural history studies where we work to understand the disease better. But also we have patient and family advisors that give us input, as Dr. Behr said, starting even before we bring a possible therapy into clinical trials and right on through the process. And regulators very much appreciate direct input from affected people and patient groups and so forth. The FDA, MHRA, and others, hold meetings in order to gather this type of information so they can make better decisions about the basis to approve new drugs. Thank you also to SADS, to Anne and the SADS team. These webinars are fantastic. We've had the honor of doing this several times. It really is a wonderful format. I know it's a lot of work in the background. And then thank you in particular to everyone who has joined today, taken some time out of your day to learn more about genetic diseases and gene therapies. We're all here for you. So please do take the opportunity to ask your questions in the chat as we go. We'll have a time at the end to go through a good number of questions. Okay, next slide, please. So this is a regulatorily required statement. It basically says predicting the future is hard, and we're not always right. I am an employee of Tenaya Therapeutics and a shareholder as well, as a disclosure. Next slide. So gene therapy for these conditions, hypertrophic cardiomyopathy and ARVC, are investigational. We are using genetic material with the hopes of halting disease progression and potentially even reversing the condition once it's established. This is what these therapies are designed to do and can do in animal models. And we are now in clinical trials to determine if we can achieve similar goals in people as well. The main feature of these therapies is they are directly addressing the underlying cause of the disease, which is a genetic variant or a genetic mutation, synonymous words. In this case, we are inserting a new copy of the gene, a functioning copy of the gene, into the muscle cells within the heart that restores the normal proteins in that cell so it can function normally. We hope to demonstrate meaningful improvements in heart rhythm, heart function, and thereby improve symptoms and quality of life. These gene therapies are given as a single dose. So we just deliver the gene to the heart, and when it's there, it stays there. And this is administered through an intravenous or IV infusion. We get this question a lot, so we highlight it right up front here. This only treats the person who gets the infusion. You cannot pass the gene therapy on to your children. Next slide. There are different types of gene therapy. Today we're talking about the first, or gene replacement therapy. This is where we are adding a new copy of a gene to people who have a dysfunctional copy, that are born with a dysfunctional copy. So this is gene replacement or gene addition. Other types of disease or other diseases caused by other types of mutation require different approaches, essentially turning off a problematic mutation. So to turn off, you can either do gene silencing or gene editing. And these both of these techniques have promise as well. They are not as relevant for PKP2 or MYBPC3, but certainly can be relevant for other forms of ARVC or hypertrophic cardiomyopathy. But our focus today is on gene replacement. Next slide. So to replace the gene, we have to get it into the heart. And to do that, we use a vector to deliver, and our vectors are adeno-associated viruses, which we'll abbreviate as AAV. So through evolution, viruses have been perfected to deliver their genes to tissues in the body, including the heart. We take out all the disease-causing parts. We take out the genes that the virus needs to divide and thrive, and we replace it with the genes that we want to use. So the replaced gene is called the transgene. In the cases we're talking about today, the transgene is PKP2 or MYBPC3 for ARVC and HCM, respectively. But we take advantage of the outer packaging of the AAV. This is a protein capsid. It's like a box that we can deliver the gene in. And they are very efficient because of evolution. They're very efficient at doing this. So we remove, as I said, all of the viral genes. So the virus itself is inactivated. We put in the transgene, the therapeutic gene we want to deliver, and then we infuse that into the patient. Next slide. So it is a single one-time intravenous infusion. As I mentioned, this viral particle or vector with the therapeutic gene will circulate through the bloodstream. And we use a vector that has a natural tropism for the heart. So it's like a box for a delivery that has an address label where the address is the heart. So those particles go into the heart to get to the heart muscle cells, the big striped pink shape here. And those muscle cells have a mechanism to bring that package inside, open it up, and use the DNA that we're delivering, the gene that we're delivering. Next slide. All right, so the gene in DNA, the RNA, and the protein are all important parts of gene therapy. But for many of you, it probably brings you back to your science class back in school. So you've heard of DNA, RNA, protein, but how they all fit together and how you should think about it when thinking about gene therapy is less than obvious. So we've built an analogy here based on furniture. So I will run through the analogies. It's 2 slides, and I'll go through the furniture scenario first, and then I'll come back and say how the scenario is similar to gene therapy. So first, the problem statement is you're hosting a party and you don't have enough chairs to go around your tables to seat everyone. But you decide you want to build more chairs yourself, but you need instructions to do that. So you order an instruction manual, which is boxed and shipped to your house. You bring it inside. Next slide. So that was step 4. You bring it inside. Step 5, you find the particular instructions that you want for your chair, and you don't want to mess up the original, and you want your friends to help. So you actually make multiple copies of those instructions, share them with your friends. Your friends take the instructions and the parts and build the chairs, and then at the end you have enough chairs to host your party. So that's the overall scenario. Let's go back 1 slide and say how is that analogous to gene therapy? So in this case, the chairs are proteins. They're specific proteins that are required for the function of the heart. But because of gene mutations, patients do not have enough of that protein. So step 2, we want to deliver instructions to the heart cells to make more of that protein. So the instructions are the gene made of DNA, and the delivery box is that viral capsid, the proteins that we've taken from AAV virus. So it's packaged and delivered to the house. In this case, the house is your heart muscle cell. So the packaging has a delivery label, the heart muscle cell recognizes it and brings the package inside the house. Now, the cell knows how to open that package and take the DNA or the instruction manual out. So this is all a process which we call transduction, the delivery of the DNA into the cells of interest. That's transduction. Next slide. Part 5 is mRNA expression. So the mRNA is the actual copies of the instructions that the cell actually uses to build proteins. So when the cell takes it up, it actually takes up the DNA. It makes many copies of that DNA in a similar molecule called RNA, or messenger RNA. So those are the instructions. And when we do the gene therapy, we follow to see how many copies of these instructions, how many mRNA copies are being made. That proves to us, when we see those being made, that the instructions have been taken up in the right cells and are starting to be expressed. All right, and then obviously the instructions are being used to build the furniture out of the existing parts. In this case, the existing parts are amino acids from protein. So you've all heard how important it is to take up protein in your diet. Your body breaks up those proteins you eat into individual amino acids, which are the protein parts, and then rebuilds proteins with those. So you already have all the parts you need. Really what you needed was the instructions to make this all work. And then with these pieces, the heart cells can complete the production of PKP2 protein or MYBPC3 protein and restore normal function of the heart cells. So I hope that analogy was helpful and it is valuable as you follow the progress of gene therapies to see, is this gene therapy delivering the gene to the heart, DNA levels or transduction, and is that being expressed in terms of mRNA and protein level. Next slide, please. All right, so there have been multiple successes in the gene therapy space, including more recent ones not yet listed on the slide. I won't go through everything, but it has been very encouraging over the last several years how many diseases have been successfully treated with gene therapies that are now available for clinical use, both in the United States and in Europe. Next slide. So there are, of course, potential risks when you receive an AAV gene therapy. A predominant one is your own immune system attacking the gene therapy. So the immune system thinks of this as foreign, as potentially an infection, and it seeks to destroy it. And that can cause inflammation, that can cause damage to some organs in some cases. So in this, to prevent this, most all gene therapy programs use prophylactic immunosuppression. That means we treat patients with drugs that turn down the immune system before we give the gene therapy, and that's intended to prevent these inflammatory reactions. The inflammatory reactions can cause adverse effect on tissues. The liver, in particular, can be very affected, and this reaction can also prevent the treatment from working. So the prophylactic immunosuppression is an important part of gene therapy. Now, at the current time, people can only receive AAV gene therapies once. The immune reaction to the gene therapy has a memory component that then makes the immune system even more robust at inactivating gene therapy if you get it a second time. As a field, we are all working on the best way to redose gene therapies, but more work is still needed here. The AAV vector that we use delivers genes to the heart, but it also delivers to other organs as well. And so we have a variety of mechanisms to reduce any potential complications of this off-target delivery. And of course, AAV-based gene therapies for the heart, including the ones Tenaya are working on, are still under investigation, and not all of the potential risks are known at this point. Next slide. So one of the features of an immune response to a gene therapy or to an AAV is the generation of antibodies that recognize it. And some of these antibodies neutralize or inactivate the vector. So just from the literature, about 20% of adults have naturally occurring neutralizing antibodies. These AAVs do occur in nature. Many of us have been infected by them. They don't cause disease of themselves, but they can cause the generation of these antibodies. And these antibodies will then block the vector from delivering the gene to the heart. And therefore, it's important to check a patient's level of neutralizing antibodies before using gene therapy, either in a trial or in clinical use. And after you have received a gene therapy, your body will develop these antibodies, and that's why you cannot receive a second dose. All right, we'll transition here into the gene therapy trials that Tenaya is sponsoring. Next slide, and then the next slide. So at Tenaya, our mission is to transform and extend lives through the discovery, development, and delivery of potentially curative therapies, such as gene therapies, targeting the underlying cause of disease. Next slide. And one disease we're very focused on is ARVC, and we are working to directly address mutations in the PKP2 gene that cause ARVC, as Dr. Behr described. This is the most common genetic cause of ARVC, although there are also several other genes that cause ARVC that we and others hope to address in the future with different gene therapies. RTN401 gene therapy only addresses PKP2 caused ARVC. We have two studies with patients ongoing now, a natural history and seroprevalence study, where we're seeking to learn more about the natural course of the disease and about the prevalence of these neutralizing antibodies that I just described. So we've enrolled nearly 200 participants overall across many countries, including 22 patients there among you in the United Kingdom. Key findings to date is that there's still, as you know, no need to tell you, still a substantial unmet need in ARVC, and people with PKP2 mutations continue to experience high burdens of arrhythmias and reduced quality of life, despite the standard treatment that's available today. We've also learned, fortunately, that most of you will be eligible to receive gene therapy because you have low levels of neutralizing antibodies. In fact, out of these nearly 200 participants, 93% of patients have low enough levels, defined as a titer less than 1 to 40, so that you meet the eligibility for our current gene therapy trial. Unfortunately, still a majority of patients are experiencing ventricular arrhythmias, with 83% of participants in this trial having more than 500 premature ventricular contractions or early abnormal beats every day. And this level is associated with a higher risk of dangerous heart rhythms. And the other trial that is active right now and is just newly active in the United Kingdom as well is the RIDGE-1 phase 1b study of the TN-401 gene therapy. We dosed the first patient in November. We've now completed the first cohort of 3 patients receiving the dose. This dose is 3E13 vector genomes per kilogram of body weight. We have gone through an external review committee, people that are not directly involved in the development of gene therapy, but are experts in gene therapy in general and in heart disease and genetic heart conditions. They reviewed our data primarily to assess the safety of proceeding with the trial, and they agreed that it is appropriate to continue the trial as planned, both by continuing that dose level, 3E13 vector genome per kilogram, and to doubling the dose to 6E13 vector genomes per kilogram. So going forward, patients participating in this trial have to have the opportunity to participate in either dose level. As a company, we have announced our plans to share initial data from this trial, particularly from that first cohort, before the end of this year. So watch out for that. Next slide. So here are some highlights of who is eligible to participate in the clinical trial. So we are only enrolling adults at this point, 18 to 65 years of age. You have to have ARVC by the standard criteria and a pathogenic or likely pathogenic, meaning disease-causing mutation in the PKP2 gene. You have to have a high burden of disease defined by having over 500 premature ventricular contractions per day. You have to have a defibrillator implanted, and you have to have, for the reasons I described, low levels of neutralizing antibodies. The goals of this trial are primarily to address and maximize the safety of the TN-401 gene therapy, to explore its efficacy, both in terms of DNA, RNA, and protein, as I described, and in terms of signs and symptoms of the disease. That will take longer and more patients, and to compare the 2 different dose levels that I described. Now, the eligibility criteria, I just want to highlight, these are not all of them. There's an extensive list designed to maximize the safety for the patients in the trial and maximize the chance that they can directly benefit from participating in the trial. So it would be very important to talk with your doctor and with the trial investigator, the doctor leading the trial at that site, to determine whether you might be eligible for this trial. Dr. Behr, I see you come on camera. Did you want to add a comment?
Just for the next slide on our where we are with the trial in the UK.
Absolutely. Perfect. Next slide, please. So what to expect if you're considering participating in a trial. So happy to announce that Dr. Behr and his site at St. George's Hospital is now active for the RIDGE-1 trial of the TN-401 gene therapy. And we hope to activate 3 additional sites in the United Kingdom in the coming weeks, both in London and in Glasgow. And the process for a patient that chooses to participate is first signing an official consent form. We don't do anything in a clinical trial without explaining the trial to you, the potential benefits and the risks, and getting your explicit agreement to participate before we do any activities. The first activity is screening and make sure that you meet all of the criteria, that inclusion and exclusion criteria for the trial. And then we do pre-dose activities to get some baseline assessments of what your disease is like. And also to initiate that immunosuppression regimen that I described. Then patients get the one-time infusion of the gene therapy. And then there's extensive follow-up visits in the first year, monitor safety primarily and to get early hints of efficacy. And then we have a long-term safety and efficacy follow-up for the following 4 years. So overall, it's a 5-year commitment for a patient and family. Dr. Behr, please expand.
Sure. So we're very excited to have already received approval to initiate the study at St George's. We're expecting 2 sites in London to start up, Royal Brompton with Antonis Pantazis as the lead there, and Perry Elliott at Barts Hospital, and then Caroline Close in Glasgow. So looking forward to getting that all up and going. And just I wanted to add that at the time of having the infusion, patients who are going through that process will be in hospital for a day beforehand and then for just under a week afterwards, so a week in total minimum. If there are any concerns or worries that patients will be kept in longer until that's sorted out, there are going to be a number of different tests required as part of the pre-dose activities, screening, but also as part of the monitoring during hospital and during the follow-up period. And this is sort of information that we can give more, that we can give to patients if they are interested in the idea of a trial. They think they may be suitable.
Thank you. All right, next slide, please. I do want to leave time for questions. I see we're getting many good questions here, so I will be brief about hypertrophic cardiomyopathy and MYBPC3, since this is the SAD Foundation talk, but we do also have 2 active trials for our TN-201 gene therapy for hypertrophic cardiomyopathy caused by MYBPC3, a natural history study in children that is showing us there's very high unmet need in children, particularly children that have more than 1 mutation in MYBPC3. This can require a heart transplant in the first months of life. And an active clinical trial of our TN-201 therapy. As another gene therapy, very similar to TN-401. It is further ahead in the trial. So we have completed dosing in both cohorts. These are the same doses we're using in 401. And our external board, the DSMB, has reviewed all the data across both dose cohort and has approved continuing the trial as planned with more patients at either dose level. Next slide. We have shared initial data from the first cohort of this gene therapy. We presented, or Dr. Milind Desai, an expert in hypertrophic cardiomyopathy at the Cleveland Clinic, presented our initial results, many of which came from his patients. And we were happy that the safety was excellent, that it was well tolerated at the 3E13 vector genome per kilogram dose. And because of the extensive similarities between TN-201 and TN-401, we do think the learnings here extend to TN-401. We also announced that we were delivering the gene effectively to the heart. We had very robust levels of DNA delivered. We could certainly detect the expression by increasing mRNA and evidence that the protein levels were increasing modestly in this case at that dose. And then we have started to see some early encouraging trends, not definitive by any means. Patients were feeling better when asked about their functional status using the New York Heart Association or NYHA class symptoms. And some of their circulating biomarkers of heart damage improved, in particular troponin levels, which some of you may know about. Those improved, and 2 of the 3 patients bound to a normal range. And Tenaya has announced we will share more data from this trial, in particular more data from the second dose cohort, 6E13, by the end of the year. Next slide. So in summary, we at Tenaya are committed to understanding your experience with these diseases. What do you need next to help you, and then partnering with you to help bring new options forward. We focus here today on gene replacement therapy, which is a potential treatment for ARVC and HCM, depending on which gene a patient has. And we are beginning to see from the TL201 therapy that we are able to deliver a working copy of the gene to the heart. Certainly, we hope we will see increasing clinical benefit for patients in the trials over time. But of course, there are significant risks as well. So if you're considering participating in a gene therapy trial, please have a careful discussion with your physician and the investigator. And of course, we highlighted 2 gene therapy programs, TN401 for ARVC caused by PKP2 mutations, and TN201 for HCM caused by MYBPC3 mutations. And the last slide just gives some opportunities for you to ask us questions or get information from us directly. Please take a look at patient advocacy at tenayathera.com. Or these websites on the diseases, arvstudies.com and hcmstudies.com. We're also, you can find us on YouTube and Facebook, etc. as well. So let's open up for question and answers. Anne, you were
Thank you very much, Dr. Tingley, and also Professor Behr for such interesting and helpful information. You've made a very complicated subject a lot easier to understand. And it's good to know that St George's and other sites in the UK are going to be involved, going to be participating in these clinical trials. That's really good news. So we'd now like to turn to questions. If we look on the chat. Right. See what questions we have here. Just looking on the chat to see what we have here. All right. So if I so the first question we have here from Marianne. My daughter had an adrenaline test today at St George's and gave blood sample and permission for biometric testing. I think that's really just a sort of a statement, really. Yeah, so that's good to see. She also appreciated the analogy of the chairs. I think most people would have done. I thought that was very good to understand, made it very easy. Another question from Laura Waters. How can gene therapy reverse the damage done to the heart and scarring? Can we help on that one?
Yeah, so we have designed our gene therapy to prevent and reverse as much as possible the disease once it's established. And in preclinical models and animal testing, it is certainly easier to prevent disease than reverse it. But we do see evidence that there is some reversing possible. And we do hope to reverse the disease in the patients we're treating in the trials now and improve symptoms. But it's unlikely that it would be completely reversible. And the question has the key thing in here, the scarring. So as part of the disease, you can actually lose muscle cells in the heart, and they can get replaced with scar tissue. And the gene therapy won't bring those cells back. So unfortunately, patients with very advanced disease and extensive scarring, our ability to reverse that may be limited. We do not know, if at what point it won't reverse disease, and that will be something we will learn as we go through the clinical trials. So we hope to help most people. We don't know, if there is a point at which it's too late.
Thank you, Whit. We have another question from Ruth Biller. Can the positive effects of the trial also be due to immunosuppression, bearing in mind that there is evidence that possible immune responses are also discussed in ARVC, e.g. in hot phases?
Yeah, perhaps this is one that Dr. Behr and I can both answer. I'll start by saying it could be a win-win, that there's benefit both from reintroducing the missing gene and from the prophylactic immunosuppression. It is clear the immune system does play, an overactivation or adverse reactions of the immune system can play a role in this disease. I will say that the patients we've enrolled, we do monitor to see if they're in an active phase at the time of enrollment. We're not encouraging that. So when we see, if and when we see, and we'll be sharing data by the end of the year, improvements in patients, it's probably unlikely turning off a hot phase for those particular patients. But as we go forward in the disease, that's certainly something we'll keep an eye out for. But Dr. Behr, your thoughts?
I suppose this is something that will come out when we start to do randomized trials. This is where we may decide to give the treatments in a random way to some patients and not to others, just because you have to establish whether it's going to cause significant, where it's going to be significantly impactful for the patient to receive the therapy. Now, that may eventually come down the line. But it's something to think about as a possible way of filtering out what is due to the immune suppression, what is due to the virus. I expect that the immune suppression will have some effect, but I think the actual vector, the gene therapy, is going to be the vital component.
Yeah, there's another part to Ruth's question. You may have covered a little bit. Do you use the same AAV-based vector in ARVC as in Danon disease, where there was a fatal event?
Yes, that's a great question. So I highlighted the successes of AAV-based gene therapies, but there are different types of AAVs. We use AAV9, which is the same as the Danon disease program. It's also the same as Zolgensma, which has successfully treated thousands of children with SMA. That was a tragic news earlier this year, the death of a patient affected by Danon disease who participated in the gene therapy trial. In this case, that gene therapy is showing great promise for Danon disease, and they are enrolling what's called a pivotal trial, the definitive trial that should support approval of that program. They announced a tragic death, obviously for that individual and their family. Also very sad for other patients with Danon disease, because it does mean it will take longer to develop that therapy and get it to more patients in the future. I think that therapy still has tremendous promise, and the company is committed to developing it. I think a key take home for folks is that death was caused not directly by the gene therapy, but a new version of the preventative immunosuppression drugs that we've been talking about. So the company was attempting to make gene therapy even safer by altering the immunosuppression drugs that they were using. And then there was an unanticipated side effect of those drugs when combined with one particular drug, when it was combined with the AAV gene therapy. So it was a particular type of complement inhibitor, a C3 inhibitor. So we don't use C3 inhibitors. when this happened, I will say that the companies are working closely together to share information. We all want to maximize safety. That's good for everybody. So we reached out to our network of people that are directly involved in that trial. They agreed we are still doing state-of-the-art appropriate immunosuppression in both of our trials. So yeah, the goal in that study was to improve the safety. Unfortunately, there was a tragic consequence. They've stopped using that C3 inhibitor, and we do not use it.
Thank you. We have another question from Cliff. Assuming trials are considered a success, how far away are we from FDA approval?
Yes, we very much look forward to the time when we have FDA, MHRA approval and other countries throughout the world. The timing will be different in different countries, largely driven by the requirements of the health authorities. FDA, MHRA, EMA do have different opinions about what they need to see before approving a trial. We as a company partner with them to align beforehand and then make sure the trials are designed to give the data that they will need to see. So we're in the process of having discussions. We do it ongoing throughout development. And we look forward to aligning with health authorities, and eventually we will announce what that alignment is, what the plan is, what data we will need to show before an either accelerated approval or a full approval of our gene therapies. But short answer, that's still a few years away. Completing and getting approval is still a few
here. Can gene therapy reverse any damage or just stop progression? Can it be given to prevent any progression for a gene-positive child that has no symptoms? The lady says here, I have the PKP2, but my two sons, aged 7 and 4, have had no gene testing done yet.
Yeah. So I'm sorry that the disease is in your family. I hope your children will be spared and that gene testing will be normal. As I mentioned, when there's extensive fibrosis has already occurred, we're not likely to be able to reverse that. We are very effective at preventing disease in animal systems. But our first trials are in patients who are already affected by the disease. If we show robust efficacy and a robust safety profile, the ultimate vision is to treat earlier in the course of disease to move more into a prevention mode. But we do want to establish the best dose and the best immunosuppression regimen to use before we treat patients early in the disease course.
One more question here. Any thoughts or forecasts regarding gene therapy development for DSP mutations in ARVC in general, and also for affected adults carrying affected babies in utero?
Yes, so we are not working on in utero delivery, but it is something that the field is considering and working on. Yeah, DSP, another devastating gene mutation causing ARVC. It is, as you point out, too long to fit in our vector. The AAVs all have a limited carrying capacity, and that's a longer gene. So we and others are working on approaches for this. We know there's a big need for this. We hope to bring something forward. The general theme is two different vectors with different halves of the gene in a way that the cell can put those two pieces of instructions back together into one. So it is more complicated. It is less efficient in animal models. And so those are the hurdles that we are working through.
Thank you, Dr. Tingley. I also thank everybody for all their questions that we've received. We'll put these to our speakers and to our SADS advisors and get back to everybody individually if we haven't covered the questions that you've asked. So it just leaves me to say thank you very much for joining us. and everyone who listened and everyone who participated. Also, a big thanks to our speakers and, of course, to Tenaya Therapeutics for sponsoring the event. Thank you so much.
Thank you, Anne, and thank you again, everyone, for joining.